which I think you guys did, not in last lab, but the one before that. So, I just kind of answered the next question. You inoculate an agar slant with inoculating loop, and you just kind of swipe it back and forth across the top. And then in agar deep, you just stick the inoculating needle down into it. What is a contaminant?
Isn't it just unwanted microbes?
Yeah, it's just like an unwanted microbe in your culture. And so we have, let me think. So we have 4 different mediums. What mediums are bacteria most easily transported in?
Is it the slant?
The slant and also the broth culture. Because the broth culture, you can culture like just tons and tons and tons of bacteria. So that's why sometimes they like to use broth. And the Petri dish, not so much, because it's like you'd have to stack them on top of each other and have this big tower. So Petri dishes are most just useful for just like looking at bacteria and just like looking at their shapes and, they're different, like if they're filamentous or like if they're ubanit, the different types of bacteria. So, okay, let me see. Any questions on lab 4? So I was thinking about asking this. Do you think that he's going to ask about these different types of growth on the slant? It seems like that's a little.
One that we had to look at.
Did we?
I don't know. I don't remember.
I don't remember either.
I know the turbidity one, but I don't.
That was for broth.
Yeah.
So in your book, there's different types of growth on the agar slant. I don't know if he's going to ask you specifically, like if he's going to have a slant and say, what kind of growth is this? But you might want to just look that over. It seems like that might be a little too detailed for him, but
It's better to know than not to.
yeah, better safe than sorry. So I would look at, in my book, it's on page 37. So just kind of be familiar with that. Okay. And he's not going to ask you about like specific bacteria and whether it's.
Yes.
Oh yeah, not yet. Whether it's like gram positive or gram negative or, well, actually gram positive and negative he might. You think?
Yeah, I don't think you need to know the names, but I think you have to know how to detect if
Yeah.
you're not negative.
Yeah. Okay. So moving on to lab 5. Any questions on lab 4? Good. Okay, lab 5. How and why do you fix a sample on a slide? It's pretty simple. But I know he asked about this on one of our quizzes, so I would know. I would remember how you fixed the sample. So in this lab, it was with heat. And the book talks about chemical fixation, but since we didn't do that in lab, he's probably not going to ask you about that. So I would know just about heat fixing and why you do it, which it fixes the sample to the slide so it doesn't move around. And then in lab 5, this is just where we did the simple stain. And so you guys should know the difference between a simple and a differential stain. Can anybody tell me the difference?
Come on, you guys know this.
Simple is just like staining it one color,
Yeah,
right?
just one stain.
And then the differential stain is like the gram positive or the acid.
Right, it's where you use like 2 different stains.
Yeah.
And different bacteria will retain a certain stain. Some will retain the primary stain and some will retain the counter stain. And let's see. So this was a pretty simple lab. I would go over this and just kind of remember how you inoculated the slide. So you used inoculating loop, and for the solid medium, first you put the water on the slide, and then you put a little amount of the culture onto the water, and then kind of mix it around. So the liquid medium is easier. You just put a drop of the liquid medium on there, and then you allow them to air dry, and then you fix them. You heat fix them. So just be familiar with that. And then as far as the simple staining goes, put the stain on, rinse it off with water, and blot it dry, and then look at it. It's a lot more simple than the gram stain. Okay, any question on that, on lab 5? Okay. So lab 7, gram staining. So this, I know you guys talked about in lecture 2, so this should be kind of a review. Okay, what is the difference between a gram-positive and a gram-negative cell wall?
Peptidoglycan.
Right, peptidoglycan is in the gram positive. Yeah, I know, see, it's a small thing, but you gotta remember it, gram positive. And is a gram-positive cell, when it's stained, is it blue or is it red? Blue. So blue is positive. For gram staining, blue is positive, red is negative. And I wish I had like an easy way for you guys to remember that. Just red, bad, red, negative. I don't know. That's the only thing I could come up with. Okay, why are understanding cell wall components clinically useful? So when if you guys are going into the medical field and you have patients come in and you actually, culture maybe like their throat, you might actually just do, I don't know if like a nurse would do it or they would send it to a lab, but they would do the stain on the spot. And, it only takes a few minutes and you would look at it, and if it's gram positive, then what could the doctor do immediately?
Penicillin.
Right, penicillin, because penicillin interferes with peptidoglycan synthesis. Whereas if it stains gram negative, then you know that you have to do some more testing to figure out what it is. So that's pretty much, you just have to get a firm understanding of peptidoglycan, penicillin interferes with peptidoglycan, and gram positive will be really affected by that. Okay, so gram staining technique, you guys should be really familiar with this. Okay, what is the first step of the gram staining process? What dye do you use?
Crystal violet.
Right, crystal violet. And then what comes next?
Iodine.
Iodine, which acts as a mordant. So it just kind of makes it really stick tight to the peptidoglycan in the cell wall. And then what's step 3? What comes after that?
Ethanol.
Right, ethanol. So if it's a gram-negative bacteria, it won't have the peptidoglycan, so it'll just, all the color will wash right out. And then the secondary stain, or the counter stain, is safranin. Yeah, and so that should dye everything red. Okay. Yeah, so just be able to know, because sometimes he might ask something like, what is the primary stain in the gram staining? So just know that it's crystal violet. The counter stain is safranin. Iodine functions as the mordant, and ethanol is the decolorizer. And I know in the lab, so technically, between each step you do wash it with water, but these steps, they're kind of just implied. The only one that, like if you were going to write out the steps, you would just do the water at the very end, washing it off. Just so if you're going to write it out, you just go directly from crystal violet, grams iodine, ethanol, safranin, water, and then you look at the bacteria and see if it's blue or red. And positive, blue or violet is considered positive. Pink or red would be considered negative. Okay. Any questions on gram staining? Okay. So next is acid-fast staining. So let's talk about the difference between acid-fast and non-acid-fast cell walls. Can anybody tell me what is, so in gram-positive bacteria, there's peptidoglycan in the cell wall that retains the stain. So what's in the cell wall of an acid-fast bacteria that retains the stain?
It's that waxy
Right.
something.
It's a lipid, mycolic acid. Yeah, exactly. Right, and so the carbonyl fusion kind of, for some reason it has an affinity, like with the lipids, and it's able to penetrate. Of course, you have to let it sit there for 5 minutes first, so that's what attaches to the mycolic acid. And, okay, what 2 diseases are caused by acid-fast bacteria?
Tuberculosis
Mm-hmm, that's one.
and leprosy.
Leprosy, yeah.
So if it is acid fast, it does have the mycolic acid.
Exactly.
And if it isn't acid fast, then it doesn't.
Right. And unfortunately, acid-fast is just the exact opposite of gram staining. So a positive acid-fast, something that does stain acid-fast, is red. And if it's not acid-fast, it's blue. So I wish I had a good way for you to remember that. I just know that it's just the opposite of the gram stain. So just get the gram stain straight, and then just remember the acid-fast staining is the opposite. Okay, and then let's see, let's go over the steps of the acid-fast staining. It's not as long as the gram staining. So the first one we start out with is carbonyl fusion, which is red. That sits on there for 5 minutes, and then the decolorizer that we use for acid-fast staining is, anybody know? Does anybody tell me? Acid alcohol. Yeah. That's what it says in the bucket. Right? Acid alcohol. And then what is the counter stain? Methanol blue. So it's a little bit shorter than the gram stain. Okay. Anything else you guys should mention about acid-fast staining? I feel like I'm missing something. Oh, in our book, I don't know if you guys noticed, but it talks about how you would put the carbonyl fusion on and then heat it up. We didn't do that process. We did something called the Quignon modification, which is a cold stain. I don't think you're going to have to remember that name, but just remember we didn't heat up the carbonyl fusion in our lab. We just did the cold stain process.
I don't know if this will help, but what we did is we made an acronym of the steps.
Oh, okay.
So I don't know if that would help you, but just do the first letter and then make a funny sentence or a funny word that you would remember.
So like you start with like C, like carbonyl fusion or something?
Yeah.
Okay, and then the next
It was
step.
Schwam.
What was it?
Schwam.
Schwam?
Yeah, because it was carbonyl fusion, heat,
Oh, okay.
saffron. Or what else?
No, acid alcohol.
Acid alcohol.
And then the counter stain.
Oh, it was wash. That's why it was brown.
Oh, Schwan. Okay. All right. Yeah, in the book it does say you do wash it off after each step.
He never
But-
had that.
Yeah, but the only one you really need to know is the last one, the last water. You do, so you do carbonyl fusion, acid alcohol, methylene blue, water. And I don't think they need to know heat because we didn't even do heat in the lab.
It just
in-
made Schwam.
Yeah, in the lecture you might need to know the heat step, but as far as the lab goes, I don't think you need to know that. So, acid alcohol.